The body has a natural way of degrading proteins. And as our proteins age, we want to replace them with, new, fresh proteins. And the ubiquitin ligase pathway is one way in which our body will get rid of proteins within the cell and recycle them. So a PROTAC is a drug that binds to a protein and basically flags it such that this pathway will recognize it. And ARV-766 is a PROTAC that binds to the androgen receptor and labels that as being ready for disposal. And one PROTAC can take out as many as 400 androgen receptor molecules. So this is a way that we can control patients or cause cell death in patients who have resistant prostate cancer. And this has been shown in the laboratory. ARV-766 binds not only to wild-type androgen receptor, but mutations of what's called the ligand binding domain, which is the front portion of the androgen receptor molecule where testosterone or other ligands bind and therefore activate it. So the purpose of this trial was to evaluate ARV-766 in those patients who had previously been treated with chemotherapy, and they could receive up to 2 prior chemotherapies, or next-generation hormones, they could receive up to 3 of those, and to determine whether we could see any effectiveness and also, of course, safety. And what we found was that patients who received ARV-766 had a 43% rate of a 50% PSA reduction. And those patients who had measurable soft tissue lesions had a 30% rate of reduction by RECIST criteria. So there is activity of this particular drug in this group of patients. Of course, the question is safety. And the majority of side effects that we see were nausea and fatigue. We did have 3 toxic deaths on the trial. They were deemed to be unrelated to the drug. So disease progression as well as other causes were seen. So the drug is relatively safe, and it's also effective. So our plan is to move this drug into phase 3 studies. It has demonstrated activity in castrate-resistant prostate cancer as well as a acceptable safety profile.